It's a pleasure to be here. I have the privilege of representing Team Project 6, which is a part of the CNRX AViDD Center, headquartered at Stanford University and run by Jeffrey Glenn. So we all know that viral RDRPs represent a very tractable target for antiviral therapy. In the cocktail used to cure hepatitis C is the polymerase inhibitor sofosbuvir. These nucleotide substrate analogs can function by different mechanisms and ultimately either terminate RNA synthesis or cause mutagenesis or stalling of an elongating polymerase once embedded within the RNA. The coronaviruses actually encode a 3' to 5' exoribonuclease that can actually remove nucleotide analogs from the viral RNA and thus get around many of the traditional ribonucleotide analogs that are out there. So what our vision is in Project 6 is to try to counteract the antagonism of the proofreading exoribonuclease by using one of two approaches. So one thought is to be able to create nucleotide analogs that can resist excision by the exonuclease. And then a second approach is to identify inhibitors of the exoribonuclease itself so that we can potentiate the activity of these nucleotide analogs. So the group is divided into 3 areas with 3 central objectives. The first is the group working on nucleotide analogs, trying to identify those that and determinants that will interfere with excision, but trying to identify just more broadly, more potent antiviral nucleotides in general. The second objective is to identify small molecules that interfere with the activity of the exonuclease. And then we have a team of individuals that work on biological efficacy and resistance. So this is an international collaboration. Riboscience is in Palo Alto and actually is providing all the nucleotide analogs. Here at UNC, we're using enteroviruses as our primary target for both biochemistry and for biology. In Amsterdam, we have David Dulin and the technology he brings to characterize a lot of the nucleotide analog inhibitors. At University of Connecticut, we have Sandra Weller, Dennis Wright, and Lee Wright developing the small molecules that inhibit the exoribonuclease. And then for biological characterization, we're working with Yan Xiang at UT San Antonio and Rolandino at UCSF. So I'm going to summarize the major achievements of on the antiviral nucleotide side of the project first. We've discovered 26 nucleotide analogs that have broad spectrum activity, meaning they are effective against both enteroviruses and SARS. And these initial screens were done biochemically. Of these 26, 21 actually failed to be utilized by mitochondrial RNA polymerase, and POLRMT is the primary off-target for many of these ribonucleotide analogs. So that indicated the potential to perhaps create safe nucleotide analogs. And of the 18 of the 21 screened, 10 were poor substrates for the exoribonuclease. So we did have some indication of substituents that would antagonize excision by the proofreading activity in the coronaviruses. And all of these were efficiently phosphorylated. So for the initial evaluation, we used biological evaluation. We used poliovirus and enterovirus. And the reason for this is that we had reporters that allowed us to produce nanoluciferase upon infection. And the generation time of poliovirus is 6 to 8 hours. So these are pretty quick experiments relative to the SARS experiments, and they don't require the BSL-2 or BSL-3 environment. Sorry. So what you can see here is replication of poliovirus in the absence of drug, and then in the presence of different concentrations of remdesivir. And you see the inhibition. So we were using remdesivir to set a baseline for the assay. This is a linear transformation of that data, and you can see at 6 hours, we can get a linear response. So we have used the 6-hour time point with poliovirus to do all of our dose response analysis. And that yields for remdesivir an EC50 on the order of about 0.4 micromolar. So we have screened a variety, through the library, the library that was produced by Riboscience to identify multiple inhibitors of poliovirus that functioned at the single-digit or better micromolar range. And what I'm going to tell you about now is the most potent compound that we've identified thus far, which is this 1251 that has an EC50 on the order of about 4 nanomolar. So with all of the molecules in the library, we're looking at derivatives that contain substituents at the 1', 4' position. And we're adding substituents to the 2' position as well. And we're looking both, in the ribose configuration and in the ERA configuration. So these are the data for 1251. You can see that it is able to efficiently inhibit polio with an EC50 in the nanomolar range, and this inhibition is not related to any toxic effect. This is an ATP glow assay. Yan's lab has been able to look at the effect of this compound on SARS-2 using a variety of assays. And again, the outcome is that we see an EC50 in the single-digit micromolar range for SARS-2, but this is substantially higher than what we observe for polio. What we know from the biochemistry is that this molecule can be excised by the exonuclease. And so we suggest, the current hypothesis now is that the substantial difference in the EC50 is related to the ability of the exoribonuclease to excise this molecule. So I'm going to tell you a little bit about the mechanism of action. So we know that right now there are 3 classes of mechanism for antiviral ribonucleosides. Mutagenesis is one, and molnupiravir finds itself in this class. There are chain terminators, obligate and non-obligate, depending on whether or not there is a 3' hydroxyl. We reported a few years ago that the pyrazine carboxamide, favipiravir, actually uses a backtracking mechanism to misregulate recombination as a 3rd mechanism of action. And so a lot of these studies have actually benefited from the collaboration with David Dulin, where he's developed an assay that uses a magnetic tweezer to monitor polymerase activity. So you have, in this case, a templating strand tethered to a magnetic bead, and to that templating strand, a primer is annealed. The polymerase can assemble and elongate to form double-stranded RNA. And under constant force, as you convert the single-stranded template into double-stranded RNA, the distance from the surface changes. And that distance is monitored by monitoring the diffraction pattern under a microscope, and you can do this for hundreds of beads. And so in doing these experiments, one can look at processive elongation by the polymerase. So what I'm going to show you first is the data that was published by David and others on remdesivir that changes somewhat, I think, the way that we have to think about the mechanism of action. So these are the control experiments showing processive synthesis by the SARS-2 replicase. If you add an obligate chain terminator, a 3' deoxy nucleotide, you can see that you are incapable of making full-length RNA. But when you add remdesivir, which is thought to be functioning as a delayed chain terminator, you can actually see that there is pausing that's occurring, but there's restart of most of these chains. And so you can actually make full-length RNA in the presence of remdesivir, but you delay the time it takes to make full-length RNA, which is likely destructive for and catastrophic even for virus replication. Looking at these, taking a closer look at a lot of these curves revealed to us that the enzyme was moving back, forming a backtrack state, and a structure of such a state has been reported by Liz Campbell and Seth Darst at Rockefeller, where the 3' end of nascent RNA is partitioned now through the nucleotide channel, and it leaves the RNA binding site that's used for elongation. So with 1251, we've performed experiments using the tweezers, and interestingly enough, you can see that we're making pauses that can restart, but when we take a real close look at these data, we are not seeing backtracking like we see with remdesivir or favipiravir. So this suggests that perhaps the 1251 molecule represents a fourth mechanistic class of antiviral ribonucleotide. Okay, so in this first part of the talk, I've told you that we've identified several nucleoside analogs with antiviral activity equal to or greater than observed for remdesivir. The most potent molecule, 1251, has an observed EC50 in the single-digit nanomolar range against poliovirus. The activity of 1251 against SARS-2 is much reduced relative to poliovirus, which is likely a reflection of excision by the exoribonuclease. And incorporation of 1251 monophosphate into viral RNA induces pausing by a backtracking independent mechanism, thus defining a fourth mechanistic class of antiviral nucleotides. And so we're going to be doing more related to that in the future. So now to switch gears and talk a little bit about the exoribonuclease. So studies performed in my lab published recently have established assays for the NSP10, NSP14 complex. The association of NSP10 and NSP14 to form the active exoribonuclease is very slow in vitro, but once formed, there's a huge interface and a very, very high stability of the complex. The complex binds RNA. It's not a very processive complex. It's able to remove 1 or 2 nucleotides per binding event, consistent with the proofreading activity of the enzyme. The hypothesis driving co-transcriptional proofreading that we've developed is shown here. So we know that the polymerase can make many errors. The replicase itself alone, once an error is incorporated, really does not change its position relative to the 3' end. it's likely that one role of the 5' to 3' RNA helicase encoded by NSP13, it may be to move the replicase out of the way when a mistake or an analog that can be sensed is incorporated. And because of the size of the NSP1014 complex, this is drawn to scale, you can see that the exoribonuclease itself is as large in dimensions as the replicase. And so we don't envision proofreading like a DNA polymerase 3 type of mechanism where you are just toggling between active sites within a subunit. We think that there's probably going to be some type of physical movement, a push and pull type of a relationship for proofreading in the exoribonuclease. So all proofreading exoribonucleases use a very similar mechanism, a 2-metal ion mechanism where you have 2 magnesium ions there to guide binding of the substrate, to activate the water nucleophile, and to also stabilize charge that builds up in the transition state. And so that's illustrated here. Activation of the water to become a nucleophile leads to cleavage. And this can be inhibited if one creates molecules that will interfere with substrate binding, but that take advantage of the high affinity that can occur when you have your small molecule binding to the bound divalent cations. So this has been the strategy that the UConn group has used, a 2-metal ion mechanism antagonist, and they're using hydroxypyridinone derivatives based here that are able to bind to the 2-metal ions. To do a lot of the screening and characterization, we have been taking advantage of an assay that uses 2-aminopurine. So 2-aminopurine alone has a high fluorescence when embedded in double-stranded RNA, as in a prime template that would be the substrate for the exo. You can see that the fluorescence goes down substantially. So excision of the 2-aminopurine terminated RNA leads to a huge change, increase in the fluorescence. And that's kind of shown here. For all of the analysis that we have been doing, we have been using an NSP1014 fusion protein. That protein is isolated from bacteria, seems to be in an active conformation, and you can see we can use 10-fold less of that enzyme than the complex that we've studied previously. So we have succeeded in identifying a variety of compounds. This is again the work of the UConn group. I'm showing you just very few here, but we've done cell-based assays and identified molecules that have single-digit micromolar activity against the exoribonuclease, and much of the biochemical data is being collected now and is consistent with those activities, which is placing us in a position now to begin to ask the question directly of whether or not we can see potentiation of antiviral nucleotides like 1251 by co-administering these exo-Wnt inhibitors.